Welcome to today's Explainer, everyone. Take a seat, get comfortable. I am absolutely thrilled you're here today because we are diving into what is, without a doubt, the single most important AI breakthrough in modern biology. We are stepping right out of the traditional textbook and straight onto the absolute cutting edge of biological research. We're talking about a leap in technology that is literally rewriting our understanding of life as we speak.
I want you to imagine something for a second. What if you could bypass a year of grueling, painstaking lab work and just save 5 to 10 minutes? Now, for decades, that idea was pure science fiction. But today, it's reality. It is a massive leap in human capability, and it fundamentally changes how we interact with the very building blocks of life. So let's jump right into this.
Here is our quick syllabus for today's Explainer. We'll start with the protein origami machine. Then, the 100K bottleneck. Third, we'll hit the AlphaFold AI breakthrough itself. Next, some really surprising discoveries. And finally, designing synthetic medicines. Okay, section 1, the protein origami machine. Let's do a quick refresher on the biological foundation of what proteins actually are and how they're built, just to set the stage. So, thinking back to the central dogma of biology. Step 1, right? Your DNA is essentially this 1D instruction manual. It tells the cell how and when to build proteins. Step 2, a machine inside your cells reads those instructions and starts linking chemical groups together, amino acid by amino acid. Now think of this intermediate stage like a long, floppy rope, maybe 300 links long. But here's the catch. A floppy rope cannot do any complex work in your cells. It's useless until it becomes step 3, which is a fully formed, highly specific 3D nanomachine. So how exactly does that 1D rope become a functional 3D machine? Well, it builds itself. Some parts of this rope are greasy, right? They're terrified of water and want to hide on the inside. Other parts have a positive charge, some have a negative charge. And because of these fundamental physical properties, the rope autonomously crunches and folds upon itself. It forms these intricate helices and sheets, packing tightly into a compact 3D object. It is, quite literally, autonomous protein origami. Which brings us to section 2, the $100,000 bottleneck. Because there was a huge problem. See, while reading the DNA, getting those 1D instructions became incredibly cheap over the years, figuring out the actual 3D folded shape of that resulting machine remained agonizingly difficult. The old manual way, it required enormous experimental facilities. We're talking synchrotrons the size of small villages. It could easily take a full year of intensive lab work, which, by the way, frequently ended in total failure. And it cost roughly $100,000 just to map one single protein structure. Enter AlphaFold. This deep learning neural network can predict that exact same structure directly from the DNA sequence in just 5 to 10 minutes, and it does it with near experimental accuracy. Let's put that into perspective and look at the sheer scale of this. 200 million. That's a staggering number. Before AlphaFold, humanity had mapped about 140,000 protein structures, and that was after decades of global collective effort. AlphaFold has now predicted the structure of about 200 million proteins. That is essentially every protein from every single organism whose full genome has ever been sequenced. The bottleneck wasn't just widened, folks, it was completely shattered. All right, moving on to section 3, the AlphaFold AI breakthrough. Because I want to make sure you all realize this wasn't just some sudden overnight magic trick. Machine learning is a really painstaking craft. When we look back at major scientific breakthroughs, we always tend to assume it was this smooth, linear climb upward. But the lead researcher, John Jumper, shared a timeline that shows a very different reality. The DeepMind team operated on these intense 6-month cycles. For the first 3 months, they'd try out their wildest ideas, and the progress chart? Totally flat. Zero progress. Obviously, by month 4, absolute panic and terror would set in. But then, around month 5, an idea would finally hit, triggering this cascade of other ideas, leading to sudden, massive elation. It was just a constant cycle of alternating terror and elation. But there was this one specific moment when they realized they had truly cracked the code. The AI suddenly started performing so incredibly well that John Jumper and his engineering lead actually panicked. As the quote says, "This is really feeling too easy. Are we leaking the test set?" They were terrified they had committed the absolute ultimate sin in machine learning, accidentally letting the AI cheat by giving it the answers hidden in the training data. They spent ages frantically looking for a leak, zeroing out coordinates, double-checking everything. It actually wasn't until they predicted some unreleased COVID-19 proteins, and the physical lab experiments came out months later, perfectly matching their AI predictions, that they finally knew for sure. It wasn't cheating.
It was literally just that good. Okay, part 4, surprising deep learning discoveries. Because this is where the story shifts from the AI just doing our math faster to the AI actually teaching us fundamentally new things about biology. Let's start with a quick definition. A homomer. This is a protein complex that isn't just one single chain, but multiple intertwining copies of the exact same chain working together, like a trimer, which has 3 copies. Keep this in your mind because remember, AlphaFold, in its early versions, was only trained to look at single, isolated proteins. Because of that, researchers were utterly baffled when AlphaFold started outputting structures with these giant, empty, voided cavities right in the middle, or these floating spirals that looked completely chemically impossible. The atoms and proteins are supposed to be tightly packed, right? At first, everyone thought the AI was just broken, but the AI was returning these weird structures with extreme confidence. Why? Because the AI had implicitly learned that the single protein it was folding was actually part of a homomer in real life. Even though no one told it about the other missing copies, it left perfect, geometrically precise negative space for them. The AI actually understood the 3D context of biology better than its own training parameters. Here is a second definition we need: disordered proteins. Now, these are specific regions of a protein that literally do not have a fixed, organized 3D shape in nature. They are just naturally chaotic and flexible. So when researchers ran AlphaFold on random human proteins, it would sometimes spit out these long, ugly, arcing ribbons. Again, the team thought, oh great, it's making a mistake. And the AI's confidence scores for these ugly ribbons were incredibly low. But then, a researcher on the team cross-referenced these supposed failures with biological databases. The aha moment was staggering. Those ugly ribbons perfectly matched known disordered regions. AlphaFold's lowest confidence score wasn't a failure at all. It was actually a state-of-the-art detector telling the scientists, hey, I can't fold this because in nature this part doesn't fold. The AI was discovering biological facts through its own perceived errors. Mind-blowing, right? Which brings us to section 5, designing synthetic protein medicines. Let's look at what scientists are actively doing with this superpower right now. We are shifting from just experimental discovery to active synthetic design. Out in the real world, scientists have already used AlphaFold to help map the nuclear pore complex. That's the giant gatekeeper of your cell's DNA, a structure far larger than AlphaFold was ever even designed to handle. In another case, researchers were trying to figure out exactly how egg and sperm fuse during fertilization. There's one missing protein link. Instead of testing 2,000 possible proteins in a lab over several decades, they just ran them all through AlphaFold. The AI pointed to exactly one protein that fit perfectly. They knocked it out in the lab, and boom, fertilization stopped. Decades of biochemistry established in a fraction of the time. But honestly, most fascinating for the future is synthetic protein design. Scientists are designing entirely new, non-natural proteins for medicines, and they found that if they run their new designs through AlphaFold first and only physically manufacture the ones the AI thinks will fold correctly, they get a 10-fold increase in their success rate. AlphaFold has literally become the ultimate filter for synthetic biology. And that brings me to this incredibly powerful quote from John Jumper himself. DeepMind built the tool, and they absolutely deserved the Nobel for it. But as he says, I'm waiting for the second-order Nobel of someone who used AlphaFold and their own creativity to discover the next thing. I firmly believe that in 20 years, nearly every person on Earth will likely benefit from a diagnostic tool or a drug that was influenced by this specific AI. It is now a foundational tool of modern biology. So, my students, I want to leave you with this final thought. The bottleneck of simply seeing the microscopic world, it's gone. The blindfold is off. We can now accurately predict the shapes of the biological machines that make up the known living world. So the question for your generation is no longer, what does it look like? The question is, what will you design with the blueprint of life? Thank you so much for joining me on this explainer today. Keep your curiosity sharp, and I'll see you all in the next one.